to call Dr. Oshin Dhillon from India, from Department of Zoology, Akal University in Punjab, to present the growth promotion and immunomodulation with Autochthonous probiotic bacteria in Labeo calbas. Yes, please. Is Dr. Dhillon with us?
Yes, ma'am. Can you hear me?
Yes, we can hear you.
Okay, ma'am. I'm sharing my screen.
Yes, please do.
We know is
Yes. Can you put the camera on? We do not see you.
I have some issue with my camera, so that's
Uh-huh. Okay. Okay. Please. 10 minutes is yours.
okay. Thank you, ma'am. Greetings of the day to everyone. I am Dr. Oshin Dhillon from Akal University, India, and thank you for the opportunity. It's my pleasure to present a part of my doctoral work entitled Growth Promotion and Immunomodulation with Autochthonous Probiotic Bacteria in Labeo calbasu. So, as we know, the scenario of the increasing population, the escalating food demand and nutrition security has just enhanced the need of the demand of animal protein, and that can be equated with it can be satisfied with the intensive aquaculture, not only to enhance the fish production but also to quench the appetite for the animal food. So in this aspect, the nutrition and disease-free environment are essential because they will help in the growth and development of the species in question. So therefore, to the culturing of the fish species, the knowledge of the optimum protein requirement is very much essential, and to find a very cheap source with all the protein containants in it, it is also very important. But we generally find that if we are incorporating some plant protein source, there is always some kind of deficiency in the essential amino acids. So for compensating the deficiency of the amino acids, we can supplement the crystalline form of the amino acids, but then also it can increase the cost of the food production. And on the other hand, for providing the disease-free environment, there is a risk of emergence of diseases with the commercialization of aquaculture. Although the antibiotics have proved a lot in eliminating the pathogens, yet they can result in the antibiotic resistance. So in this context, the probiotics have gained much attention. And commercially available immunostimulants are also there, but probiotics isolated from the gut of the host itself, that means the autochthonous probiotics, they can be helpful. So keeping in these conditions in view, the present work has been done on Labeo calbasu. Labeo calbasu is the state fish of Haryana, and it also fetches a great market value due to its unique taste. But its population in the natural water bodies, it's dwindling due to human intervention. Although some ranching programs have been established by the government, yet this is not enhancing the population in the natural waters. So a next step can be the fish culture in semi-intensive systems so that it can contribute to the intensification of the present species. So the present work has been taken into consideration, and the candidate species has been selected. So it was done to evaluate the effect of autochthonous probiotic, and it was compared with the other feed supplements in Labeo calbasu fingerlings. So for the present work, the fingerlings were procured from Hoshangabad, Madhya Pradesh, and they were brought up to the laboratory where they were acclimated for a few days. And finally, the feeding trial were taken for 90 days. There were 4 treatments. Treatment 1 included soybean-rich diet, which was at the optimum protein requirement, and that was 40%. Thereafter, in T2, we incorporated duckweed, and as duckweed is deficient in lysine and methionine, so we have incorporated crystalline forms of lysine and methionine in T3. And the incorporation of Bacillus anurinoliticus, this is the autochthonous probiotic strain. It was incorporated into the diet of the fish in T4. So this table, it shows the ingredient and proximal composition of the experimental diets. This was the experimental setup. Various growth parameters and the intestinal enzymatic activities were determined following the standard protocols. The biochemical assay was determined, and the fingerlings, they were finally challenge trial with a common fish pathogen, that is Aeromonas hydrophila, after the feeding trial of 90 days. And before the challenge trial and after the challenge trial, hematological diagnosis was done. And at the end, when we found the most possible results, that particular specimen was further processed for the immunohistochemical assay, which was just to visualize the adhesion of the probiotic bacteria that was incorporated in the diet. So these were the results. The growth performance was significant in T4 fed group, where the supplementation was done with the autochthonous probiotic, and the growth performance was high in terms of specific growth rate. The feed conversion ratio was low. Feed conversion ratio, it depicts that the proper utilization of the food material takes place, and if the value of feed conversion ratio is low, that means the higher specific growth rate would be observed. Then on the proximate carcass composition, it was observed that highest quantity of protein were found to be accumulated in T4. This table shows the total number of the erythrocytes and leukocyte post and pre-challenge with the Aeromonas hydrophila, and it was observed the significant count in T4, which depicts the immune response of the probiotics fed. Now this figure, it shows the intestinal histological section of the gastrointestinal tract, and the lower two figures, figure this one and this is depicting the presence of bacteria, probiotic bacteria, and they were immunolabeled, and they have found positive results. So the bacteria which was incorporated in the diet of the fish, it was found to adhere inside the gastrointestinal tract. So this was the reason why so much growth was observed in the fingerlings of T4. So we have at last the salient findings of the study. Duckweed, which we have incorporated in diet second, fourth, and third, it was found to be eco-friendly, and it is easily available at farmers' doorstep. It is not only pocket friendly but also environment friendly, as it does not contribute to that much quantity of the ammonia and phosphate in the holding water. Therefore, the supplementation of lysine and methionine can be cultivated by the Bacillus anurinoliticus, which not only provides enhanced growth but also contributes to the immune response. Survival of the fishes after the challenge trial was found to be optimum, and the presence and colonization of bacteria was also observed. Therefore, this particular feed formulation can help in the manufacturing of the feed for the Labeo calbasu. So if we are actually conserving the fish species at a smaller level, that will enhance their large production also. Further, they can be incorporated, they can be added up into the natural water body, so that the population of these fish species can be protected. So the present study, it aims at the conservation and protection of the fish species. I would like to acknowledge the department and the University Grant Commission for the financial help. Thank you, everyone.
Thank you very much, Dr. Dhillon, for this concise presentation. Very